Introduction. Welcome everyone to our presentation. Today we will be discussing our research on the differential modulation of aquaporin 1, referred to as AQP1, in chronic lung toxicity models through the exposure of various toxicants on mice. This study was done in collaboration with teams from Georgia University School of Medicine, University of Cincinnati Medical Center, Nova Southeastern University College of Osteopathic Medicine, and the University of Virginia School of Medicine. Background. Aquaporins, also known as water channels, are membrane proteins that facilitate the movement of water through lipid membranes by means of osmosis or hydraulic gradients. The expression of aquaporins can be affected by environmental toxicant exposures, which can result in lung injury. Inhalation of toxic chemicals or particulates disrupts lung homeostasis, causing pulmonary edema, inflammation, and/or other lung toxicity effects. Our study focused on three types of toxicants: particulate toxicants, such as carbon nanoparticles, referred to as CNT, and chemical toxicants, such as cigarette smoke extract, referred to as CSE, and the combined effects of these two toxicants, CNT plus CSE, to assess their impacts on lung aquaporins, mucosal defense proteins, and markers of pulmonary injury. Methods. Our study used a strain of C57BI-6 mice, which were exposed to either CNT, CSE, or a combination of both in a subchronic toxicity mouse model. The oropharyngeal exposure regimens in mice were designed to reflect real-world exposure. We assessed the expression of aquaporin 1, mucosal defense protein, SP-A, MUC5B, and markers of lung injury through techniques such as quantitative reverse transcription, PCR, and histopathological analysis. Results. Our results demonstrated that aquaporin 1 expression was significantly upregulated in CNT-exposed lungs while it was downregulated in CSE-exposed lungs. In the combination model, CNT plus CSE, the aquaporin 1 expression showed a mixed response. The differential regulation of aquaporin 1 suggests that it could be a critical molecular marker for identifying specific types of lung injuries related to different environmental toxicants. We also observed a significant downregulation of mucosal defense proteins, SP-A and MUC5B, particularly in the CSE and combination exposure groups. This downregulation points to a compromised homeostasis in the lung, likely leading to increased susceptibility to infections and other complications. An analysis of the bronchial alveolar lavage, referred to as BAL, fluid protein content indicated increased permeability and infiltration in the lung tissue, especially in the CSE and combination groups, further highlighting the extent of lung injury induced by these toxicants. Histopathological analysis revealed significant changes in lung tissue, including bronchial alveolar hyperplasia and increased infiltration of immune cells, especially in the CNT and combination exposure groups. Resulting conclusion. Our study demonstrated that different toxicant exposures, whether particulates or chemicals, brought about distinct patterns of lung injury and modulation of aquaporins. These findings highlight the potential of aquaporins and defense proteins as biomarkers for lung injury and how they can be used in understanding the pathophysiology of toxicant-induced lung injury. The downregulation of mucosal defense proteins further demonstrates the harmful effect of toxicants on lung health. We hope these insights lay the groundwork for future research that aims to develop potential drug-based therapies for individuals who are affected by lung toxicants. However, more studies are needed to fully understand the role of aquaporins and their application in clinical practice. Acknowledgements. We would like to thank the University of Cincinnati Center for Environmental Genetics for supporting this study through an NIH award. Thank you for listening in on the effects of toxicants on lung health and the role of aquaporins.